Welcome to our podcast on the endocrine system. Here we will cover some of the same slides that we saw in our lecture podcast, but then we will add to that, expand upon that, so we can look at the structure of many of these endocrine glands, including the pituitary, the thyroid, parathyroid, and the adrenal cortex. We will see some images of pancreas and pancreatic islets, but we won't go into detail about the cells of those. So here we have the pituitary gland, and we know that the pituitary gland is going to be divided up into the nervous-like portion and the glandular portion. So as we look at this, we can very quickly determine that we are looking at this portion that is more corded. You can see more cells, greater density, and this is going to appear very neuronal-like. So this is going to be our posterior pituitary. This is our pars nervosa, part of our neurohypophysis, all these different names that come into play. And then here is the adenohypophysis and the pars distalis portion of the adenohypophysis, or the anterior pituitary. And here we have the pars intermedia. These are really the only parts that we're going to be able to see here. We're not going to see the pars tuberalis portion of this one, the infundibulum, those things. But this is our adeno, and this is our nervous portion. Now again, the adenohypophysis, when we're looking at an H&E stain, we're going to be able to visualize the really dark staining basophils versus our acidophils versus the chromophobes, and we saw this one from our lecture podcast. And in these, often it's going to be very difficult. We can see some lighter cells, the chromophobes, that you might see here, possibly a lighter staining one here. There's not a lot of chromophobes in this particular image. Then we see the very red acidophils, and then we're going to see the others that aren't the red staining, and these are the more bluish stain, and these are going to be the basophils. The basophils are going to make up the bulk of what we see here, but we have the basophils versus the acidophils versus the chromophobes. And again, this is going to be making up our adenohypophysis. The neurohypophysis, this is going to be very neuronal looking. The cells we see, we collectively call them pituocytes. And as we look at higher magnification a little bit later in our slide presentation, remember that the mottled look that we're going to see, especially if we look in this area, we can see some fairly large ones. These are called herring bodies. These are accumulations of the hormones in the cytoplasm of these pituocytes. We're also going to have capillaries, lymphatics in through this tissue. So this is a slightly higher magnification view of our pars nervosa portion of our neurohypophysis. And again, higher magnification, we can see that model look. These droplets of tissue that we see, that's going to be the herring bodies in that portion of the neurohypophysis. Now here we have a slightly different view, and these different stains are going to pick up different cells. We're not going to really visualize what's happening in the neurohypophysis, but this particular stain, when we look at the pars distalis portion of the adenohypophysis, it will be able to allow us to see so much better the different chromophils that are going to be present in this tissue. So as we look at this particular side, we can see a lot of very dark staining cells that are present in this tissue. And with this particular stain, all of the dark staining cells that we see are going to be our basophils. And these are going to be present here in this tissue. We're not going to be able to identify the cells that aren't staining. Those would be the acidophils and the chromophobes. But with this particular stain, we can see the abundance of the basophils in this particular tissue, part of the adenohypophysis. Now, this is going to be the look in the neurohypophysis. just a very busy look. But these are all the herring bodies that you can see with this particular stain in this tissue, and it's hard to make out even the nuclei of these cells because the herring bodies in this neurosecretory material is staining so darkly in this tissue. Now, the stain for acidophils is going to look very red. And once again, when we look in here, we can see some of these really dark granules, and I'm not sure that they're really the acidophils. The acidophils are going to stain more homogeneously through here. And as you look, there are fewer cells that are present that are going to have this staining for the acidophils present. So again, the majority of the cells are going to be basophils. We will see the acidophils, and then lower number are going to be the chromophobes. Now, as you can tell, we have shifted gears, and even at this low magnification, I'm hopeful that you can appreciate the organization of this tissue, and you can recognize the follicles that are present. Don't really get drawn into looking at the color. The stains might have different colors. Look more at the organization, but hopefully you can see this and very much know that we are looking at thyroid tissue. Again, this is going to be one of the easier ones to really pick out. At low magnification, you sort of have a gut feeling that that's going to be the case. But then at these higher magnifications, you can clearly see our principal cells surrounding a thyroid follicle. You can see these all very regularly arranged. We have our colloid material on the inside. And then as we look at some, we're going to see these extra cells, very dark staining. They're going to interrupt the pattern of our follicular cells on the outside. Here we see some, and in this one, I think we're going to look a little more closely. We see the interruption of these cells. These are the parafollicular, or the C cells, that are present surrounding our follicles. So once again, we have our follicular cells, our principal cells, and then in this particular one in the very center, you can see the disruption of that organization. And these are the parafollicular, or C cells, because these cells are going to be producing calcitonin. Now, if you see an image of thyroid, and these follicles are much larger than what we saw in the previous slide, and they may change in their size. So again, don't be restricted to a certain size, a certain color. And in this particular slide, the principal cells are going to be more purple staining, and the colloid is more pink than what we saw in the previous slide. So again, don't get drawn to color. But now we have this glandular looking material, something we sort of lean toward a gland as soon as we see it. And so this is going to be our parathyroid gland section through adjacent to the follicles. And remember, parathyroid sounds very similar to parafollicular, but they are two distinct groups of cells, two distinct positions. The parathyroid is an organ, whereas the parafollicular cells are within the thyroid organ itself. So let's make sure to keep those separate. So here we're looking at the parenchyma in our parathyroid. And as we look at the parenchyma, remember, we are going to have two cell types. The majority of cells that we see in this field, these are going to be our chief cells, but then we're also going to have our oxyphil cells. And those are going to be present. These are the much larger ones that we see here. These are our oxyphils, and the remainder are going to be our chief cells. Now, we didn't talk about this in our endocrine lecture, but I want you to appreciate that when you're looking at a tissue, and again, at first glance, it's really difficult to tell an organization. You see a lot of dots, and so you might at first think lymphoid tissue, but it's not organized in such. You may think thymus, but again, we don't see Hassel's corpuscles. We don't see a cortex and medulla. But then your eyes are drawn to these areas of light staining patches. These are our pancreatic islets. That's going to be the key that we're looking at pancreas, and these are our pancreatic islets, or islets of Langerhans. Now, the bulk of the pancreas is exocrine in nature, but the islets are going to be the endocrine portion of the pancreas. And when you look more closely at these are going to be clusters of cells. You have alpha cells, beta cells. There are a number of different cell types that we are not going to characterize, but certainly cells are producing insulin from these pancreatic islets, among other hormones that are being secreted that help with metabolism, that help with digestion, that help with the movement of glucose. Certainly, that's the role of insulin in and out of the cells. Now we're going to take a look at the adrenal gland, and the key feature of the adrenal gland is that cortex, that trilaminar cortex with the central medulla. That's going to be your key, that you are looking at the adrenal gland, or the suprarenal gland, as its term anatomically would be. Just giving you a little closer view here, we can see the trilaminar appearance to the cortex, and then we have the medulla on the inside. So we're going to continue to go up in magnification, continue to look at these 4 layers, 3 cortical, the 1 medullary. On the surface, here we can see our subcapsular space. This is where we're going to have that plexus of blood vessels that stream down through the cortex. But here again, we can see our cortical trilaminar. We've got the zona glomerulosa, the zona fasciculata, the zona reticularis, before we get to the medulla. And we'll look at those more closely to really get an appreciation for the histological characteristics of each. Now, what we're going to do here, let's just go back. We're going to look at this region, and you can see it's somewhat laying on its side. We're not going to go up and down. We're going to go from right to left as we go down through these layers. And so here we have the capsule, here we have our subcapsular plexuses, and this is our first layer of the cortex. And as you can see how these are arranged almost in an acinus, it just looks like little florets to me. As you can see, the petals, sometimes they'll have something in the center. And this is the zona glomerulosa. Glomerulus is going to be around, it's sort of a synonymous term with an acinus. I like glomerulus reserved for the kidney, however. So this is the zona glomerulosa that we see, preceded by the second layer of our spongiocytes, and that's the zona fasciculata. And that's what we see in this image. Most of the time when you're looking at these sections, it's going to be comparative. So you're not just looking necessarily at such a high magnification of one to really be able to predict and to look at and to determine exactly where we are relative to other areas and other features. So for instance, here's a higher mag, and you may not at first be comfortable going, yeah, it's an acinus of something, but I'm not sure. Well, here this almost looks like connective tissue, but then you see these spongiocytes, and that's going to be your clue, the associative nature of one to the other. That is so characteristic of what we see in the adrenal cortex that you will know based on that arrangement that we're looking at the zona glomerulosa followed by the fasciculata. That makes it much easier. So keep those associations in mind, and don't just focus on the characteristics of a particular region, but also know what are the adjacent regions and how would that help me determine where I am. Now, the fasciculata and the spongiocytes, they are so classic, so characteristic, it's going to be difficult to really confuse this. About the only thing I think you could possibly put this with as far as a look-alike in histology, if this were just a standalone slide and you weren't exactly sure what system we were talking in, you may confuse this with a multilocular adipose tissue or brown fat. But again, typically in the human body, we're not going to see that much, that vast percentage of brown fat. Also, you can see these somewhat sinusoids running through and how these are arranged in plates or columns. Again, that's very characteristic of what we see in the zona fasciculata. So putting all of those things together, the rarity of the multilocular adipose tissue, the arrangement in columns of this tissue, your sort of differential diagnosis for this histologically would just naturally be looking at the zona fasciculata and the spongiocytes. Now, here's another area that you really need to see the associations one for another. If not, then you're just sort of looking at parenchyma. You sort of think that it's secretory epithelium, but you're just not sure. So as you see the spongiocytes, the fasciculata melding and changing and transitioning into these cells, notice these are no longer arranged in little bundles, acini, glomeruli. But again, these are going to remain in somewhat this columnar chorded appearance. We'll see this a little better as we step back and look at a higher magnification of this. But as we move from the fasciculata, we're moving into the zona reticularis, or the network of cells that we see here. This is going to be our third of the deepest cortical layer adjacent to the medulla. So here, as we look at the third layer, we're transitioning away from cortex into medulla. We can again see the reticulated nature, this networked nature of our parenchyma, our glandular epithelial cells, and this is our zona reticularis. And then we move into this tissue, and you can see these large nuclei with a central nucleolus, very neuronal looking in nature. So we know that this chorded nature, this almost, if you didn't know better, almost looks a little bit hepatic, like a liver. That's how I like to think of it, but still distinctly different from that. And we move into this tissue, then you know we're looking at adrenal cortex, reticularis, versus a medulla. And in the medulla, and again, this is a situation where you really wouldn't know what we're looking at here unless you had seen a lower magnification slide or saw an inset of a lower magnification slide. But these are just the chromaffin cells present in the medulla that secrete mostly epinephrine. I believe 85% is epinephrine, 15% is the norepinephrine.